Okay, by now you know the drill. Empty room, closed door, thus no mask. So let's talk about energy transfer, the energy of life. This biker is extracting energy from the food he's eating, using it to power his muscles. But what is energy? Energy is defined as the ability to do work or to move matter. You have two different forms of energy. You have kinetic energy. Biker on the hill has high potential energy. Coasting down the hill, the energy is released as kinetic energy. So kinetic energy, energy in motion or energy of movement. Potential energy, stored energy available to do work. If you ever watch college football, for instance, on Saturdays, and they'll talk about a particular player and they'll show they're 6'2" and they weigh 247 pounds, and they're a psychology major. One of the very common majors that you will see in college football nowadays is something called kinesiology. Kinesiology is the study of movement. It's the kinesiology majors is what historically would your PE teacher in high school would have had. You had a PE teacher, they would have had a degree in physical education. Well, that's been changed over the last 25 or 30 years, and those folks are now getting a degree in what is referred to as kinesiology. Again, it's the study of movement. It comes from the word kinetic. Here you have a compressed spring has potential energy. You release that compression. That is kinetic energy, and you see it's throwing a tennis ball in the air. The energy we get comes from bond energy, from chemical bonds. Chemical bonds have potential energy. This glucose molecule stores energy in its chemical bonds. To release the energy, the cell breaks the bonds. So the bonds break here, energy is released, and you wind up with, instead of 1 6 carbon structure, you have 2 3 carbon structures. And in the next chapter, we'll talk about what happens to these 3 carbon structures. Or maybe we'll talk about it in this chapter. Let me not get ahead of myself. Energy changes within a biological system. Energy is never created nor destroyed. It is only converted. That is the first law of thermodynamics. Here we have a triphosphate or a 3-phosphorus product, triple adenine triphosphate. Here's the adenine molecule or adenosine molecule. And here we go back to our producers and consumers. Here's the producer, makes the sugars and potential energy. The deer eats the leaves, releasing some of the energy released is heat. Other is you get chemical energy, and the deer is able to run and jump and hop and do all the things a deer has to do. Energy from the sun cannot be used directly by cells. It must be captured, stored, and converted before it can be used for cellular use. The process that captures, stores, and converts it is photosynthesis. Photosynthesis takes the energy of the sun, converts it into other products. Hang on just a second, the back is coming off of my clicker. Okay. Photosynthesis takes the energy of the sun and carbon dioxide and water and produces sugar. So it takes the energy of the sun and 2 inorganic molecules, converts them to an organic molecule, and then that is the potential energy. Then that potential energy is then broken down by the deer eating the leaves to release some heat and other products. Energy release is really inefficient. We will talk at some point about how much ATP can be produced. And what we're going to talk about is a theoretical number of ATP molecules that can be produced. Usually that number is going to be significantly less. But the energy of heat production is very disordered. It cannot be converted back to a useful form of energy. Once you burn a log, there's no way or you've burned a piece of paper, there's no way to spontaneously put that piece of paper back together again. It's required. Energy must be there for life to exist on Earth. Cells are constantly using energy to build their molecules and structures, perform chemical reactions, and carry out life's processes. Again, same little model, always energy input, and you wind up with what you need to get life. Entropy is a measure of disorder. The randomness of the universe can be described as disorder or entropy. I had a professor over at UALR that was really loved him some entropy. He talked about entropy all the time. But since heat energy is constantly being lost to the universe and heat energy is disordered, entropy of the universe is increasing. This is the second law of thermodynamics. Basically what it says is that every time energy is used, some of that energy will be lost to the universe as heat. And the example picture they show you here is an intact light bulb. It's very highly ordered, but you smash the light bulb and now you've got all these shards of glass and everything else that's now highly disordered. So this is the intact bulb. You drop it, exert some energy to break it, and that breaking increases the amount of disorder or the entropy. So, what does life on Earth depend so heavily on sunlight? Why does Earth depend on sunlight? Energy, the capacity to do work. Energy can either be kinetic or potential. Energy in motion, energy stored. Molecules, held together by chemical bonds, transformed from one form of energy to another, converting the potential energy to kinetic energy. Metabolism. Metabolism is a word we've all heard. We all know that one person in our life that can eat anything they want to eat, never seem to gain a pound, and we always say, man, that person has a really good metabolism. Well, metabolism is both energy in and energy out. When we think about breaking down food, that's our one side of our metabolism, but then taking the products that we break down in our food and converting them back to fats or proteins or whatever, that's the other half of metabolism. So metabolism is all the chemical reactions that go on in your body to rearrange atoms. You build complex molecules out of simple ones, like the monomers we talked about in chapter 2, to form new chemical bonds. We have 4 individual structures. You put some energy in, you now have 2 pairs. You put some more energy in, you now have a cube of 4. You take that same cube of 4, energy out, you wind up with the two atom molecules. A little more energy, now you've got your 4 individual units back again. So energy in can build things up, energy out breaks things down. Reactions form bonds that build molecules and their energy input. The inputted energy is stored in chemical bonds. Reactions break chemical bonds to release the energy. This picture here is showing some men making what looks to be a barn. Looks like they're having a barn raising. Consult your history instructor about barn raisings. But you see, what they're doing is they're bringing pieces together. You know, each one of these little trusses is anchored, so they'll bring one in, anchor it to the one next to it, et cetera, et cetera. That's what you're doing here. You're just putting energy in. You know, they're swinging hammers, using a drill driver, whatever they're doing to put them together. And then down here we have a building that I hope is the result of what is referred to as a controlled implosion. That's not a terrorist bomb that has done that. But this is where they have a very large building, and rather than taking it down a little bit at a time, they cut some main members at the bottom, and then they put explosives in, and they blow the building up, and it collapses in on itself. Again, energy was being released. A lot of energy in those rocks, the concrete and glass and metal and everything coming down. Lots of energy being released. Some chemical reactions transfer electrons. We talked about the transfer, the movement of electrons in chapter 2. We talked about those different types of bonds, the ionic bonds and the covalent bonds. Here in an oxidation reduction reaction, you actually have an electron changing place. In the oxidation reaction, you have the electron moving. So here you see you have an electron donor molecule. It's got 2 electrons in it. Oxidation occurs, and now those 2 electrons are gone. In the reduction reaction, you have an electron receptor. This molecule, whatever it is, gave up its 2 electrons. They were picked up by this one, and it's reduced. Only a chemist would call something that's being bigger and say it has been reduced. Why do we say this has been reduced? Well, if you are adding electrons, which are negative charges, to a molecule, what are you doing to the overall charge of this molecule? You are reducing it. This one's being oxidized, the electrons are leaving. This one is being reduced, the electrons are being accepted. Oxidation reactions is the loss of electrons from, or oxidation is the loss from an atom or a molecule. It releases energy. The energy released is stored in the electrons. So you have a release of energy over here and a capture of energy over here. Reduction, again, the gain of electrons. So the reduced molecule gains energy. So again, this now has this extra molecule, and if you'll look, this one, very similar to this one. So now this molecule could undergo oxidation, release energy, and then be reduced yet again. Oxidations and reductions occur simultaneously, therefore they are often referred to as enzyme reactions. There is something called an electron transport chain, which is a series of proteins within a membrane participating in a sequential linked oxidation reduction reaction. Electron comes in here, has energy, it loses that energy, is picked up here, gains energy, is picked up here, etc., throughout the end. And ultimately, the electron will be dropped at the end of the chain to some electron acceptor. Most often, what happens is a water molecule will be formed at the end. Redox reactions release a small amount of energy. Photosynthesis and cellular respiration both use electron transport chains. As energy is released, cells store it and use it in other reactions. This is being released into the cytoplasm of the cell. It is released in the form of photonic energy. They will then be collected and stored in a molecule called ATP. There will be a low energy molecule, ADP, will take this energy and will be converted into ATP, which is a high energy molecule. And then those ATP molecules are used further down the line. So what is oxidation and reduction, and why are they linked? Oxidation, loss of electrons; reduction, gain of electrons. And they are linked because their products and substrates sometimes are the same thing, and like in the ADP-ATP cycle. ADP gains energy to become ATP. ATP then loses energy to become ADP again. So they are linked that way. Cellular energy currency is ATP, which I just mentioned. You have adenosine molecule, and you have 3 phosphates, that is ATP. ATP is a high energy molecule. It has molecules stored, or energy stored, that can be used later. It releases that stored energy, and when it does, to drive some reaction, you have hydrolysis occur, and the ATP is dropped down to the low energy ADP, and then this excessive energy. It is formed during cellular respiration. We will talk about cellular respiration probably in the next chapter, if not in the next one, in the very one after that. But it basically goes like this. You eat something, say this energy bar, and breaking down the energy bar, you produce a lot of energy that's stored in your body in ATP. You then start riding your bicycle, you start to break those molecules down, ATP becomes ADP. That's driving the reactions that allow you to ride the bicycle or run the race or whatever you're doing. Then that ADP then will come through the cycle again, pick up even more energy. This is cyclical. ATP becomes ADP, which then becomes ATP again. You're releasing energy, and that energy release gives you the energy to perform muscle skills or whatever it is you have to perform. Reactions that break down ATP are coupled with reactions that require energy input. These coupled reactions, when working together, release energy from ATP. They can power a second reaction, which is again your muscles firing or whatever you're trying to do with energy at the time. ADP or ATP hydrolysis can energize molecules. ATP is an activated glucose, or this glucose is now activated because it's got that extra phosphate group on it, combined together with other molecules. You see, we have the phosphate here, we have a short polysaccharide, we have an activated glucose which can bind. As you continue to get this longer polysaccharide, you can form things like starch or protein or cellulose or glycogen or any of the other complex carbohydrates. ATP can donate its phosphate group to other molecules. Here's an example of a transport protein. You see you've got a lot of the little circles outside the cell, but very few circles inside the cell. But these things are too big. They can't just pass through the membrane on its own. They have to have a way to come through. And what you have is these molecules come in and then they get flushed out. And it's the ATP energy that actually causes this molecule to pivot from this configuration to that configuration. Then you don't need to draw a concept map because it's already there for you. But again, this is how ATP runs through the cyclical program to keep your cell working efficiently. Chemical reactions occur quickly. They do, however, usually require an enzyme. That enzyme acts as a catalyst to keep the reaction moving along. A catalyst is something that speeds up a chemical reaction without being consumed. You can take a catalyst, put it into a reaction, and after the reaction has completed, you can get your catalyst right back. One of the oldest examples of a catalyst or a demonstration of a catalyst that one of my professors at UALR told us was the story of stone soup. Now, I don't know whether you've ever heard this fable or not, but it involves a beggar coming into a small village and wanting something to eat. And he's knocking on doors saying, I haven't eaten in several days. Do you have anything? And everybody slams the door in his face and says, go away, beggar. We don't want you here. Go away. And after having several doors slammed in his face, he goes to, he finds a cauldron, big like thing the witches make their witches brew in at Halloween. He finds a cauldron, pulls it into the middle of the city square, finds some wood, puts it under the cauldron, builds a big roaring fire. Then he goes to the city well and he fills up the cauldron with water and starts the water to heat. Then he reaches into the little bag he's carrying and he takes out a little small bundle wrapped in cloth, and he unwraps the little bundle and he takes out a stone. And he places the stone carefully over into the kettle or the cauldron of boiling water. And then he reaches into his bag and he pulls out a spoon and he starts to stir it. Well, as soon as he started dragging things into the middle of the city square, people started looking through their shutters. What is that guy doing? And by the time he's got it full of water and he started to stir a little bit and he tastes it, people are coming out of their houses. And finally somebody says, dude, what are you doing? And he said, I'm making stone soup. And he tastes it again, and somebody says, well, how does it taste? And he said, well, it's not bad, but it would be a little better if I had some salt. And somebody in the crowd goes, hey, I've got some salt. And that person runs home and gets some salt. And he sprinkles some salt in his water, and they said, how is it now? And he said, well, it's okay, but it would be better if I had some onions. And somebody says, I have some onions. And they run home and get onions. And he chops up the onions and puts it in there. And then he says, well, a couple of potatoes would make it even better. And so somebody runs and gets potato, and then somebody else runs and gets carrots, and somebody else runs and gets peas, and somebody else runs and gets celery, and somebody else runs and gets an old ham bone that they have. And he puts all this stuff together and he stirs it up, and finally, ooh, that's the best stone soup I've ever tasted. And then he looks at the pot and he says, wow, I have made way more stone soup than I can eat. Would anybody like some stone soup? And everybody, of course, says, yes, they want stone soup. So everybody runs home and gets a bowl. And he feeds the entire village on the big giant pot of stone soup. And as he's getting the last of the stone soup out of the cauldron, he reaches in and very carefully takes out his rock, puts it back in the little fabric wrapping, wraps it back up, and puts it back in his bag. and says you never know when you may need to make another pot of stone soup. Well, the stone was the catalyst or the enzyme. There was not going to be any soup, but by placing the stone in there, he got everybody's attention, and that started the reaction. But at the end of the day, he got his stone back. That's what enzymes do. They enter a reaction, but they don't get consumed. So in a reaction, you have substrates, which are what the enzyme acts on. You have the reactants, which are the molecules present at the start of the reaction. And you have the products that are the things written at the end of the reaction. Now, we are all familiar with mathematic formulas where 2 + 2 = 4. Okay, in chemical reactions, we don't use the equal sign, we use an arrow. And the arrow simply means yield, the word yields. And you know, we're all familiar with yield signs. That means you're supposed to slow down. Well, in chemistry, when we use the word yield, we're talking about how much is being produced, the production. Like when the farmers are growing their crop, they say their yield was so many bushels an acre. So, in a chemical reaction, on the left-hand side of the arrow, you're going to have the reactants. That's the things that are going to be reacting. On the right-hand side are the products. Now, the products of one reaction may very well be the reactants of a different reaction. So what we have here is we have an enzyme and a substrate. They bind together in this chemical reaction. Here's the product. So we have these reactants and this product. Now in the next reaction, this one is the reaction, and these are the products. And if you look closely, this is one molecule here that is bound here, and it's 2 molecules on the end. So we start with a large molecule, and we break that down into 2 smaller molecules. An enzyme must perfectly fit the shape of its substrate. If it does not fit, as it said from those of you old enough to remember the OJ trial, if the glove doesn't fit, you must acquit. Well, in this case, if the glove doesn't fit, the reaction's got to quit because it's not going to continue. If the substrate doesn't fit, then there's no further reaction. Everything's going to stop. And the place where the substrate binds is called the active site. So again, there you go. You got your product molecules. Again, you got the substrate coming in, binding to the enzyme substrate complex, and then the 2 products are removed at the end. Again, the thing about the enzyme is the reaction doesn't change it. This and this are the same, so it can be used over and over and over. Activation energy is the energy required to get a reaction started. The best example I can tell you from a activation energy, if you've ever tried to build a fire. If you've been out camping and you build a fire and you want to burn some wood that's this big around, you don't start that burning with a match. Not enough activation energy in a match. What do you have to do? You have to lay your groundwork. You have to put some paper or some dry straw and some small sticks and get your fire started, strike your match. Get a small fire going, which has a lot of activation energy, then you can get your big log to burn. So the activation energy is the amount of energy required to start a reaction. And if you look over here without the enzyme, activation energy is high. You bring in the enzyme and it lowers the activation energy. So you're talking about maybe the fire is the match is I think what I'm trying to say here again is the activation energy, but the dry straw and the small twigs are the enzyme. The difference being you get an enzyme back. Only some chemical reactions occur in a cell at a given time. Again, excuse me, you have normal binding here. However, if you have a binding that occurs down here that changes the shape, so this can't come in, that would be non-competitive inhibition. Inhibition is going to be a way to control reaction. You don't always want these things to be forming. You don't always want chemical reactions to be going on. Something has to control those reactions, and that is called inhibition. If you look here, you have something, the inhibitor binding here, which changes the shape of this, so this can't bind. Over here, we have what is called competitive inhibition, where the inhibitor is now bound in the same place as our substrate molecule, so we can't get there. Two different types of inhibition. Again, inhibitors shut down molecules or shut down unneeded reactions. There comes a time when you have enough of a product, so you don't need to make it anymore for a while, and that's what happens here. One of these systems will take place, and you stop making whatever product it is you're making. Substrate molecules typically bind at the active site of other enzymes. Again, what if another molecule prevents it? That's competitive inhibition. Molecules that bind outside the active site and alter the enzyme shape, preventing the substrate and enzyme from binding together. That's non-competitive inhibition, or non-competitive enzyme inhibition. Again, hooking on down here, changing the shape of that. And there's competitive inhibition. Get the nice little same molecule as here, but now instead of binding here and changing the shape, we're going to bind in the same place this wants to bind, effectively blocking it from getting it where it needs to be. Temperature affects enzyme activity. One of the things we know for a fact is that the human body has a normal or an average body temperature of about 37 degrees. That is where enzyme activity is at its peak. If the temperature gets lower than that or higher than that, you see there is a precipitous drop in the amount of the way a reaction occurs. That's what happens when you have a fever, when your temperature, and if you will notice it's very subtle, but this spot and this very peak of this are not in the same place. There is actually, enzymes work best for human beings at a slightly elevated temperature. And what I mean is, while 37 might be considered normal, reactions may happen a little bit better at 37.1 or 37.2. That's why when we're doing all this testing and we're taking people's temperature, whether or not they can come into a building, the cutoff point is not 98.7. The cutoff point is actually, for most instances, 100. So if they take your temperature and it's 99.2, they usually let you in. But if it's over 100, they prevent it, because that 37.2, that's your body doing what it's supposed to do. Energy or these reactions work better at a slightly elevated temperature. Enzymes only have an optimum salt concentration. Again, salt concentrations, if you start to elevate or decrease the salt concentration, reactions don't happen well. Enzymes become distorted or changed, and they don't function. So an optimum salt concentration and pH. So when they take your blood at the hospital trying to determine, what illness you may have, one of the things they do is they do a blood panel, and one of the things they check is the pH of your blood. So, why is maintaining homeostasis crucial for cells in order to assure their biochemical reactions can be carried out efficiently? Because when you are in a homeostatic state, your pH, temperature, blood glucose, all of those levels are the same, and all of those enzymatically controlled reactions happen best in a controlled environment, in a specific environment. Substances can enter or exit cells through membranes. A cell's interior is chemically different from its exterior. It helps maintain the differences by regulating transport of dissolved substances across the membrane. Regulating what is inside a cell, that's an example of homeostasis. So you see, here's the cell, here's the membrane, and you'll see that these things are not anchored together, so some things can pass through them. Only certain things can pass through the membrane. Solutes enter and exit by different methods, depending on two factors: concentration gradients and the chemical nature of the substance. What is it? Is it a polar or non-polar item? How big is it? Does it have a charge? So here's an example of something called a concentration gradient. There are more T molecules near the T bag than the rest of the cup. You see the T molecules that are starting to diffuse out of the bag. Not any out here, lots in here. But what happens over time? No gradient, the T is diffused to the rest of the cup. Now you've got equal distance, all the same amount of T all over. You see it's continued to spread here, but in this last picture, it's evenly dispersed. Yeah. Okay. Maintaining concentration gradient requires energy. A gradient is more ordered, has less entropy than no gradient at all, more entropy. Entropy tends to increase unless some force is preventing it. So what could be done to prevent the diffusion of the T in this figure? What could you do to maintain this level not going back to this? One thing you can do, and you see people who drink tea do this all the time, they're constantly dipping the bag or lifting the bag up and dropping it in. What they're doing is actually stirring. They could achieve the same thing by stirring it with a spoon or whatever, but the bag is there, the bag is convenient, so you just dip the bag. Simple diffusion does not require any energy, therefore it is called a passive transport system. Takes place where there is a concentration difference on one side and the other. You see the little whatever these little blue, sort of light blue orbs are supposed to be. A lot of them over here, very few of them over here. So by diffusion, what will happen, if they are able to pass through that membrane, they will move from this side to this side. That is the area of low concentration. Small, nonpolar molecules can cross cell membranes by simple diffusion. Also, water can cross there by simple diffusion. The size and chemical properties of small nonpolar molecules allow them to pass through membrane's hydrophobic barrier. This is impossible for large polar molecules. This is a nice little apparatus that I think you can still buy one if you wanted to show this to your children or something, or just see it yourself. You used to buy these at Hobby Lobby in their little science area. But basically, it's just a U-shaped tube that has a semi-permeable membrane, and you make two concentrations. Usually, you want to make like a sugar concentration because sugar is pretty large. Salt sometimes will not work very well. But osmosis is a type of passive transport. It takes place when there is a different concentration of water on either side of that permeable membrane, and the water moves down the concentration gradient. So if you've got 80% sugar over here, or excuse me, 20% sugar, that means you have 80% water. Where over here, if you have 10% sugar, you have 90% water. So the water is forming the gradient, and since water is going to move, we're going to call that osmosis. And what you do with this little apparatus is you make up your two concentrations where you know one of them has a whole lot more sugar in it than the other one. You put the high concentration on one side, the low concentration on the other side to where they are equal. You know, you put a little mark with a Sharpie on your tube, and you fill the high concentration on one side and the low concentration on the other. And what you will see after a few minutes is the level on one side will actually start to move up from the other side. So the water is moving, and we're not going to count, but if you counted the number of these little molecules here and here, you will see, or the bigger molecule, you'll see that there's 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. They're still the same number. I'm not going to count the other ones because there's a whole lot more. But there's still the same number of sugar molecules as on this and this, likewise this and this, but the levels change because the water is moving to try to equalize the water concentrations. Osmosis determines plant water content in plant cells. This is when you water your plant, it looks like this. When you don't water your plant, it looks like this. Hypertonic surroundings result in a water loss. So these cells have plenty of water in them. These cells do not. So plants usually keep their solute concentration lower inside than outside, so water constantly is wanting to go into the cell. Hypertonic surroundings result in water loss, shrinkage of the large vacuole right here, causing what is called turgor pressure, causing a plant to lose turgor pressure, which is what causes the plant to wilt. Always we show red blood cells whenever we talk about hypertonic, isotonic, and isotonic solutions. This is what a red blood cell should look like. It is what is referred to as a biconcave disc. It is a disc structure that the middle is flattened, it's pinched in, not a hole like in a donut, but it's just pinched together. So you've got water going into and water going out of the cell. Here's what happens when you place it in a hypotonic solution. You've got water going in is much greater than the water going out, so the cell swells, and you can actually make red blood cells burst by putting it in a hypotonic solution. This is a hypertonic solution, and now the cell that's supposed to look like this, because it has shrunk and lost most of its water, you see these are very malformed cells, and they are formed. It's called cell shrinking. Equal solute concentrations inside and out of the cell, that is an isotonic solution. Higher solute concentration inside the cell. Excuse me. You can take a blood sample like this. You can take a human blood sample and put a drop of distilled water on it, and it will do this. A hypertonic solution, there's more outside than in, so the water's going out trying to diffuse those products. Lower solute concentration inside the cell. And again, that's where I think some students will get confused. We know that hypo means low and hyper means high, but then we've got low. Well, that's the lower concentration inside the cell. So when we say it's a hypertonic solution, we are referring to there's a higher concentration out here. Facilitated diffusion is also a type of diffusion that does not require energy. However, it does require some help. This is a transport protein. Again, whatever structures these are too big to pass through, or possibly they're nonpolar, so they can't pass through this, but they need to get in here, and they can still come through this protein structure. No energy being required. Active transport, however, requires energy to be released. When active transport occurs, when the membrane proteins use cellular energy to transport substances across the membrane, these biologically are referred to as pumps. The sodium-potassium pump is probably the most well understood of these pumps. If you'll notice here, you've got lots of the little purple orbs and very few of the little yellow squares outside the cell. You got a lot of it inside the cell. But what you want is you want to continue to get the purple stuff out and bring the yellow stuff in. Problem is, that doesn't work too well by diffusion, because if you've got more of it out here, if these things diffuse, then we want to go this way. But we want to go against the concentration gradient. So we have to pump them up. So these little active sites fill with these little orbs. The energy transfer occurs, and this closes at the bottom, opens at the top, and allows those things to float free. And then the little yellow structures come in, they bind at their active site. That causes these things to open again, pumping them in here. So it's a pump. It works this way and this way and this way and this way. Throwing stuff out, bringing stuff in. Endocytosis requires energy. This is another form of active transport. Cells can form vesicles as they engulf fluids and large molecules to bring them into the cell. Substances can be moved up or down their concentration gradients this way. You see the small portion of cell membrane buds inward, entrapping these particles. As the vesicle forms, the particles are trapped inside the cell. Then a fully formed vesicle has all the different particles inside the cell. That is endocytosis, or movement in. Exocytosis is exactly the opposite. You have a vesicle that's full of stuff that you need to get rid of. The vesicle will bind with the membrane, then it will sort of spread out and eject all of those products out into the extracellular fluid. Again, exocytosis requires energy. It's to move large polar molecules out of the cell. Endocytosis to move those large molecules in through the form of vesicles. If you have any questions, please send me an email either to my personal email, trussell@uaptc.edu, or the course messenger found in the contact link on the left-hand side of the Blackboard show. Let's see you later.